been long in the tooth and are retiring, so we're replacing them, or we're getting new talent in new areas. Dr. Signorini is one of those that we've gotten within the last 5 years that's added to our department tremendously. Just recently, in 2022, we were interviewing for a new turf grass extension position where our turf grass extension person had retired. And we interviewed several candidates, and Florence was one of them that we interviewed in that position. And she gave a couple seminars, one of which was her research seminar. And after we had heard all the candidates, we said, she'd be fine as an extension person, but she's a phenomenal rhizosphere biologist. And since we intended to hire a rhizosphere biologist, which we didn't have, we said, why don't we just hire her and an extension person at the same time? And that's how we got Dr. Florence Sessoms. I'll leave it to you. You can tell them all about how we might be able to use mycorrhizae to improve the growth of pawpaw.
Well, first of all, I wanted to thank the organizer to invite me. I'm Florence Sessoms, and today I'm going to talk about Mycorrhizal Association. But because I'm really relatively new here, I wanted to give you a small introduction about myself.
Put it closer to you or talk a little louder.
Excuse me. Hello. Perfect. Oui. Okay, perfect. Okay, so first of all, I'm from France, and I grew up exactly in Paris. This is my home city. All the little blue star is a place where I grew up. So Gare du Nord is where the street where I was. Then I had my elementary school, my middle school, my high school, and sorry, I'm going to try here. It was a university. So I moved out from Paris in 2005, where I did my PhD at the University of Fribourg in Switzerland. And during that time, I worked and I was introduced to the fabulous world of mycorrhiza. I worked on mycorrhiza establishment and understand the nutrition and the genetic regulation of mycorrhizal establishment in Petunia. And at the end of my PhD, I moved to the US in 2009, where I did a couple postdocs. First, I was in New York, in Cornell, at the Boyce Thompson Institute, where I still work on medical grow and mycorrhiza. And then lately, I moved in Minnesota, where I worked a little bit on soybean pathogen, soil nutrification, and turf grass science. And that's why I applied for the turf grass position in 2022. But in 2023, in August, I finally started my new position as an assistant professor at Florence State, where I worked on root, rhizosphere, and resilience. So I'm going to talk really dwell a little bit about the origin of mycorrhiza, and because really during my PhD I had to do a literature review on the origin, what are mycorrhiza, where are they coming from, and basically this is this paper that made me fall in love with mycorrhiza, because basically they were able to visualize fossil structure of mycorrhiza that are 400 million years old. And to showcase that to you, basically this is Aglaophytum major plant. This is the cortex of the plant, and here you can see the obviously life structure already completely done in this fossil. So I was very excited because I was working with a living fossil. So, moreover, and there was this other paper that showed that spore, the origin, so the first structure, were formed already 460 million years ago. So if you try to, it's big numbers, but if you try to understand how, and I'm sorry, here this is a fungal spore, like the fossil fungal spore, and here this is a present-day spore. And so if you try to understand, okay, yes, it's that long, but what does that mean in geological scale? Well, you see, this is where the spore were found in the sediment, The oldest fossil structure was arbuscular mycorrhiza, where you can see that really it coincides with the origin of land plant. So and this is the way, at that time, the continent were grouped together. So basically, the main hypothesis that I found remarkable was that mycorrhiza may have helped terrestrial plants to colonize land. And so remember I was showing you the fossil of Aglaophyton major. This is a schematic representation of Aglaophyton major, and you can see that this plant had no root system. There was this little structure here, and there were more to anchor the plant to the soil. So basically, mycorrhizal fungi, when they establish inside the roots, so here it's like really like carrot roots in a plate, they are going to form this very large, dense mycelium network that's going to go out of the roots and really forage into the soil. So basically, the idea was mycorrhiza was here to provide nutrient, phosphate, nitrogen, potassium, and water to these like early land. So most importantly, also another aspect of the mycorrhiza, this is so old that it also predates the diversification of plants. So basically, at the origin, you had like not a lot of diversity. You had already here the beginning of mycorrhizal spore were observed here. Then you have all the diversification of plant lineage. It was during the time that you had this very large continent called Gondwana. So basically, it helped like so the arbuscular mycorrhizal association. It could explain why the arbuscular mycorrhizal association can be observed with most terrestrial plants, because the establishment was done before the diversification of the plants, and why it can be found in most continents. So every continent will have arbuscular mycorrhiza present in their soil. So really, summary of like this topic was that it's an science by symbiosis. We are working with a living fossil. They are thought to have helped like the colonization of land by early plants, and they predate the diversification of plant lineage. And so that's why we could see mycorrhizal association with most of the terrestrial plants. Now, nowadays you have like 4 different types of mycorrhiza: the arbuscular mycorrhizal fungi, the ectomycorrhizal fungi, ectoendomycorrhizal fungi, 5 ericoid mycorrhiza that form really the species, and the orchid mycorrhiza. And for me, I'm very familiar with the arbuscular mycorrhizal fungi. I would love to work more on the ectomycorrhizal fungi because they form this like association and led to like truffle and chanterelle that I really like. So now, how do they develop inside the root system? So, as I mentioned, the spore is the first structure that are present into the soil. They are going to germinate into the soil. But the plant is going to like showcase its presence by secreting, producing some like compound, here it's trigolactin, that's going to indicate its presence. And as a result, the spore is going to germinate and branch toward the direction of the plant, but also produce another type of like effector that are going to tell to the plant, hey, I'm here, I'm coming towards you, and the plant is going to adapt to the fungus. So what's going to happen? You have this structure called aptosorium or epipodium. Then you have like a penetration inside the inner cortex and the formation of arbuscular structure. So this is a schematic representation, but I wanted to actually show you like staining and microscopic picture, because it's very difficult to see the fungus inside the roots. So what you have to do first when you harvest your roots, this is doing a clearing with KOH, cleaning the roots, and then you can stain it with like some stain that's going to attach to the fungal cell wall, and then you can actually visualize the fungal structure outside and inside the roots. So here there is the acrosoria, they can form coils, you have arbuscule here, what we call their infection unit, Some mycorrhiza form a vesicule. This is a storage place inside the roots. And then at the end you have the spore. So what I want to mention is that mycorrhiza are obligate biotroph, meaning that they need a host to have a complete life cycle. Without a host, you will have the spore germinate and then go back inside the spore. Okay? Now, I wanted really to go a little bit further in the arbuscule. The arbuscule is a site of nutrient exchange between the plant and the fungus. So the fungus, with its mycelium, is going to forage into the soil and provide water and nutrient toward the plant. And this exchange is going to occur here inside the cell. And it's a symbiosis, and so it means that you have an exchange between both partners. So as a result, the plant is going to give sugar from its photosynthesis and lipid to the fungus. Okay, so that's why it's an obligate biotroph, because they need the sugar from the plant to develop. But if you think about this arbuscule, the arbuscule structure never destroys the cell. They never destroy the cytoplasm. It's not the pathogen. You have this membrane that will form, around the arbuscule, and the arbuscule lifespan is between 7 to 10 days, and at the end, the arbuscule will degrade, and the cell can be reinfected. So here again is a schematic representation of how the arbuscule develop. You have the hyphae, a little trunk, a young arbuscule, mature arbuscule, senescence, and death. So, why it's important? This is the arbuscule again, site of the nutrient exchange between the plant and the fungus. And so high phosphate level, we are going to inhibit colonization, but not only the colonization, also the presence of arbuscule. So during my PhD, that was one of the first graph I made actually. I looked at colonization of an arbuscular mycorrhizal fungi in function of a gradient of phosphate. And you can see, so in terms of colonization, I'm looking at all fungal structure, internal hyphae and arbuscule. I could see it was decreasing with higher concentration of phosphate. Now, when you look at the normal colonization, this is the abnormal colonization at high phosphate level. So not only you are losing your internal structure, like your internal hyphae, you are also losing the arbuscule. So there is no exchange now between the plant and the fungus. So, and I'm sorry, I moved too fast. Yes, okay. So why are we using arbuscular mycorrhizal fungi beside the nutrient uptake and the increased biomass? Well, this is because in the literature, in greenhouse experiment, in ghost chamber experiment, we feel there is an incredible number of benefits that are provided by the association. And we have resistance to drugs, resistance to salt stresses, a better tolerance to cold and heat stresses, and also a plant priming. So basically, the plant, by the presence of mycorrhiza, is going to be primed, and it's going to be more resistant to certain pathogens, not all of them. Now, is it true? So the main issue is, as I mentioned, all plants can be colonized. I mean, all plants, 80% of the territorial plant can be colonized, but doesn't seem to have what we call a host specificity, but there is a certain what we call host specialization. So not all interactions are going to give you a positive result. And so this paper really showed, so this is 64 different types of species that was tested with one fungus, one mycorrhizal fungus called Glomus etunicatum. And here this is the percentage of change of biomass from the control. Mycorrhizal plant relative to the control. And you can see that here you have an increase of biomass, where here you have a decrease of biomass. And it's very interesting because it's not really a pathogen, but it will lead to a decrease of the plant fitness here. Okay? And again, this is because of this nutrient exchange. The plant, there is a cost to being colonized by the mycorrhizal fungi. So in addition to that, and I really did this from this paper, not only the plant genotype or the fungal the partnership between the plant and fungus can vary in function of the species. the presence of a biotic factor, like for example other bacteria, a bacteria inside the soil, and also the abiotic factors such as soil texture, like soil nutrient, presence of organic matter, can have an effect, an overall effect on the outcome of the association. And for us, what would be an increase in biomass, a better yield, maybe some quality, like a testing quality, etc. of the fruit. So, really in summary of like what I just said, this is arbuscule as a site of nutrients exchange. Phosphate will have a huge impact on the total colonization and the arbuscule presence. The arbuscular mycorrhizal symbiosis provide many benefits that are beside the plant nutrition, but the outcome of the interaction will depend on like the host and the fungus, but also on like environmental and microbial, other microbial factors. So now, when I was asked to talk today, I really tried to do a literature review on mycorrhizal present in pawpaw tree. You know, I was not very familiar with this tree and with this interaction, like the interaction between mycorrhiza and pawpaw. So I tried to look and do like really Google using Google and other like websites where I can find literature, and I found very few scientific papers. The only one that used pawpaw, where they were talking about mycorrhizal pawpaw, was that one, but there is no proof. They just basically took the tree and considered that this tree was in symbiosis with mycorrhiza. So, since I could not find an actual proof, meaning, and for me a proof is actually seeing microscopy staining with fungal structure inside the tree. This is for me is a way of like really saying, okay, here we really see an interaction. I could not like find it with like pawpaw, so I went and I looked at like where the family where pawpaw come from, the Ananaceae family, and after, again, by really some digging, I found this paper that have like looked online at all the different trees and different terrestrial plants and confirmed that if there was like mycorrhizal presence inside this plant. And this is one of the tables. And in this table you can see that several Ananaceae species were in fact colonized with arbuscular mycorrhizal fungi. So that was interesting. So actually look at the reference and try to find the paper. And this is a paper with like custard apple, and you can see that there is actually fungal structure in it. So this is from their paper with like the little arbuscule in it. But also when they were inoculating the custard apple, this is a control, and this is with inoculated plant. You can have an increase of the an increase in the height of the shoot and also the number of leaves. So that was great. Then I looked at another reference, and I could see both of these like species, Ananaceae species, had actually arbuscular mycorrhizal fungi. But what was interesting from these two papers is that, and again we are thinking of biomanagement practice, it seems that the Ananaceae family can only be colonized, as we know right now, with arbuscular mycorrhizal fungi, and not the ectomycorrhizal fungi. So if you are looking for, I would say, for an inoculum, I will focus on an inoculum that have arbuscular mycorrhizal fungi. Now, you know, when I was like looking at the summary of my literature review, I was yesterday, and it's really yesterday at 11 p.m. I said, I was like, okay, I didn't find any picture of like mycorrhizal establishment in pawpaw tree. And then it happened. So basically, I found from Florida State University, it's really a slide from a lab for a class. And basically here, there was like what I call my unicorn, the fungal structure inside the cell of a pawpaw tree. So with a little arbuscule, the internal alpha. So here, this is the only time that I've seen fungal structure that are stained inside the pawpaw tree. And it's really from a lab. A lab book for a class taught at Florida State University. So, scratch that. I did find a microscopic picture. Pawpaw seems to be able to be colonized with mycorrhizal fungi. Anyways, the Ananaceae family species can be colonized. There is fungal structure inside the roots, but more importantly, only arbuscular mycorrhizal fungi were shown to be associated with this species. So now, I've been told that most of you are using inoculum, like a mycorrhizal inoculum, when you are transplanting your plant, because it helps with like the plant establishment in a new soil. And so I was thinking, okay, that's great. If you think about like the short-term practice, you want some fungal presence inside the soil to make sure that the plant is going to establish and at the beginning. But I'm thinking we also think about what could be the best management practice to improve the mycorrhizal establishment with Pawpaw. So basically, you don't only want to have like mycorrhizal when you actually plant. You want to make sure that you're keeping the mycorrhizal presence during the growth of like the growth of the tree. So I would say, based on what we have discussed, we need to reduce the application of synthetic fertilizer, especially I would say phosphate, because phosphate will inhibit the arbuscular presence and the total colonization. I think we have like seen in other like orchid with almond, never with Pawpaw, so I'm basing that from like other literature review with other trees, like really reduce the organic amendment, use organic amendment such as compost, plant with the Pawpaw tree, mycorrhizal friendly species, or cover crop. So again, this is based from literature with other trees. I think there is like a lot of research that could be continued with Pawpaw in terms of what type of species could we use really to promote, what type of cover crop could we use, buckwheat, alfalfa, and also on the long term, how is this like a co-transplanting, or if transplanting or having intercropping or cover cropping within an orchard of Pawpaw will help with like the growth, the establishment, the growth, yield, and also food quality. So that's the end of my talk. I wanted to thank you for your attention. This is my email address if you have any questions, and thank you very much.
So, do you sounds like a fertilizer that's low in phosphorus would be a good idea? Is that right?
Yes.
NPK, like high, low, medium, maybe. And also, would you recommend that we spend money on some of these mixes, gallons of spores that you can buy from these mycorrhizal spores available from nursery suppliers? Is that worth the money to sprinkle that into your containers or along the ground of your orchard? Any benefit in that?
So it's I would say it's a bit hard for me to answer that question because I have never done an actual experiment to test that. I would say that the mycorrhiza is not going to affect your plant, it's not going to kill your plant. I would say yes, you can use this type of inoculum. If you are looking at an inoculum source inside like a store such as Ace, Yoder, Home Depot, or even local nursery, I will always look at the bag and see what type of mycorrhizal fungi you have and what is the concentration of spores or like hyphae that they have, because you want an inoculum that have a high diversity of fungus but also a high concentration of spore too, because the more the merrier. Also, as I mentioned, literature showed that only arbuscular mycorrhizal fungi can form symbiotic relationship with the Ananaceae family. So I will focus on an inoculum that have only arbuscular mycorrhizal fungi. Yes?
Are there any weeds or cover crops that actually kill the mycorrhiza? I have some garlic weeds in my orchard, and I was told that they're killing the mycorrhiza.
So personally, I don't know. I would say that the only species that I've known that do not form mycorrhiza are from the Brassicaceae family, so that do not form mycorrhizal. I'm not sure about garlic weed.
You mentioned planting plant-friendly mycorrhizal species. You mentioned planting, plant-friendly mycorrhizal species. Are these generic to all mycorrhizae? And if they are or not, what plants can you name that?
With Pawpaw, no. This research that had been done was on almond orchards. I would say people were using buckwheat. I know that alfalfa, buckwheat also is buckwheat, alfalfa, because also I think it can form the rhizobium symbiosis, so it also fixes nitrogen. So the main issue is at the end, you want, and this is really what we need to test. Okay, we try different cover crop. And we want to see on the long term, and when I say the long term, this is within the span of a growing season, how is it benefit for the pawpaw? Because, for example, I think, and you see that with in apple orchard, or I think with vine orchard, when the plants are young, there is a competition between your cover crop and your cover crop and your tree, and at the end it reduces the yield of the plant. So there is a lot, I think, of unknown that still we need to try to evaluate to provide you with the best management practice.
Two questions. The first would be, you have suggestion for cover crops that would be companion plants with pawpaw. Second, is there any advantage or disadvantage to using black ground cover surrounding the pawpaw trees in respect to mycorrhizal?
So that's a great question. So for the cover crop, I think we need to do more research to try to evaluate different cover crop species that would be the most beneficial with your pawpaw tree, with your variety, with your soil type and everything. So more to do. For the plastic, so this is my understanding that when you create, you increase the plant diversity, you also increase the soil health and the soil biodiversity. So basically at the end, you want to recreate an ecosystem that is beneficial for your pawpaw tree. So the problem is, I think if you do your black cover, yes, you reduce the weeds, but you also decrease the soil diversity that's going to be potentially beneficial for your tree. And also I would say for weed, and I'm talking from my turf grass perspective. When we use turf grass, sometimes as a living mulch, if you have, for example, a dense established turf grass or forage or whatever cover crop, you generally do not allow the establishment of weed because of their presence, because they are shading possible germination of the weed, or some even cover crop or some turf grass have allelopathic compound that can in fact inhibit the establishment of other weeds. So, yes.
It sounds like if I'm going to start out farming and getting ready to buy a bunch of fruit trees to plant, it almost sounds like it's best to buy from someone who has them potted and with the soil in contact already and not disturb that energy transfer that's already going on versus buying bare root products. Is that true or not true?
Okay, so to make sure, so basically, if your soil wasn't disturbed and there is certainly a chance that there was already colonization, I think if you transplant to basically reduce the transplant shock and make sure that your plant is going directly to establish with mycorrhizal fungi in your new soil, you could actually apply some inoculum on it at the base of the roots, because again, one of the most important parts when you apply inoculum is to be in contact or very close by the root system. The fungus needs to sense the presence of the roots to actually develop, germinate, and effectively infect the root system.
As far as the mycorrhizal, I know you can put it into a mix injected into the ground, hydraulic root zone injections. Is that possible?
Okay, so I'm very sorry, I'm not familiar with hydraulic root infection. So the concept, as I understand, this is basically to force, like the incorporation of the mycorrhiza into the soil. That will work if it's like really close after in contact to the root system, and if you have an active growing root system too. So I know, for example, for turfgrass, and again, going back to the turfgrass where I have the most experience, and that was we needed either to add the mycorrhiza at seeding because the plant will germinate and develop a root system, or we'll actually, when we aerate the soil, so we create holes, apply the inoculum, and close with like top dressing. But it was for a golf course. So maybe the same technique could be used.
I think it could because it is injecting water under pressure with mycorrhizal in it. So.
Yeah, so also like when I was, and I'm basing a lot of my experience of like all my lab experiment when we were, and that was, we were working really in very clean like surface and a little zone of like sand, because the idea we really wanted to have only testing only the benefic effect of one mycorrhiza, so we're really trying to work in sterile culture. When we were applying water, it was very, we have to be careful because the spore, they are not going to stay, they are going to be washed out. So I will say that when you have, and again, I'm not familiar with that, but I will make sure that they stay in the root zone and they are not washed away because again, they will be present in the soil, they're going to germinate, but without the host being present nearby, it's not going to develop really well.
Thank you.
You're welcome.
Any more questions? Thank you.